Awesome. Good morning. Good morning. Good morning. Good morning. It is Grab a Snack Day. It's the second of the three lectures on the myriad of strategies that invertebrates have to feed. That's where we're going to be spending our time today. There's a lot to consume in this lecture. There may be puns. And so let's get started. First of all, let me clear that up. You can do those later. Where were we last time? Feeding modes number one. We were talking about deposit and suspension feeding. So spending a lot of time on the bottom of the ocean thinking about the prevalence of these strategies that you may not have spent a whole bunch of time thinking about before and how widespread they are across this planet of ours, which is predominantly a marine environment. And so we talked about a myriad of strategies like the Cichliophrans, the whole phylum that we know only from the mouth parts of the particular species of lobster found in northern Europe. And so we've got filter feeders on filter feeders, suspension feeders on top of suspension feeders all the way down. What are we going to do today? Well, we're going to talk about symbiosis and grazing and browsing and hunting. Oh my. So symbiosis is often kind of overlooked when we think about feeding strategies, but in fact, if you kind of dial out and think about it, it's a lot of this concept of a holobiont, of a eukaryotic organism that is supported, dependent upon a myriad of other organisms and particular prokaryotes or bacterial symbionts. This is a really successful strategy all over the world. And we're going to kind of avoid some of the more well-known examples that you've bumped into before. We're going to focus on some deep-sea thermal vent examples and some crazy leaf-like photosynthesizing snails. We'll talk about grazing and browsing. That'll be our bridge because these Elysia snails do both. They are symbionts and also they are grazers. And then we'll work our way in. We'll kind of flesh out some of what we built up last time together when we introduced you or reintroduce you to the concept of optimal foraging theory. And we'll build that up a little bit more today as well. So, what are my hopes for today for you? That you can appraise, synthesize, and interpret examples about how grazers and symbionts and browsers and hunters can gather their nutrition and think about that in the context of ecological theory from kind of first principles of this optimal foraging theory, and then be able to interpret specific examples, novel examples, because of course as we're talking about the invertebrates, we're talking about examples and novelty that we're discovering all the time. And we'll focus in on a couple of examples of novelty today. Let's start sharing things. Let's start off with symbiosis. And so this is symbiosis being, of course, any kind of tight association between two different organisms living in close physical proximity. They don't necessarily have to be positive ones, right? But we're going to talk about largely positive ones because we're talking about feeding. So we're talking about one providing food for the other. And we'll kind of touch on what the other, the often bacterial partner gets in this. And the picture that you're looking at is a leaf sheep, or an artist's, Julia Stern's rendition of a leaf sheep that appeared in the Manufactured Ecosystems Forecasting Climate Futures exhibit that was on your campus all summer. And you can go following that QR code right down there, and you can take a look at it now today if you want, or you can zoom in on a high-resolution gigapan of the leaf sheep. The leaf sheep is like the Elysia sea slug. This is one of the ones that is living, accentuating how it lives by kleptoplasty. We'll break apart kleptoplasty later on. But of course, when we're talking about symbiosis, we're not talking about any one particular lineage. It's just that most animal lineages are invertebrates, so we have a choice, an amuse-bouche, more puns, of symbiotic examples to think about than you could ever even hope to imagine in like a vertebrate class. And these examples can be lineage specific or not, and we'll break apart some examples of that in terms of how your endosymbiont might be transmitted when we get to the deep sea thermal vent examples. When we're thinking about terrestrial things like from our own example, We're often thinking about bacteria in a gut that have a particular refuge or an area of the gut, but that's not the exclusive kind of way that you would expect to find them. And they can provide all of or additional amounts of nutrients or energy. And some of the most widely, surprising examples, at least in my lifetime as an old person, but as a newly old person, these are symbioses that we've only learned about during my life. And that's occurring at these deep thermal vents that you're becoming more and more familiar with your hands-on manipulation of data throughout the year today. So those include like suboglanid polychaetes with the trophosome. We'll break that up in a second because we're learning more stuff all the time. Shrimps that have ectosymbionts on their mouth parts and gill chambers. Think about the Yeti shrimp that we talked about last year that is basically doing the dance at the deep thermal vent because it's aerating, hydrating the bacteria that it kind of lives and feeds off of. And then we've got gastropods with bacteria in their gills. Those include the metallic snails that, again, all live at these fabulous deep sea thermal vents. Now, so back to 1977 and Bob Ballard and all those folks kind of discovering these things. There was the discovery, A, that was surprising of the density of life, but then B, it was the fact that, well, how are they living? And so when some of these first, particularly like this Riftia animals were brought back to the surface and dissected, they was discovered that they had no gut. And further dissection revealed that they had a specialized, essentially a pocket, a trophosome, where they kept chemoautotrophic symbionts, bacteria that are providing the nutrients needed for life for their animal host. And that discovery has kind of leached out into the rest of the phyla that we know about from these areas. So at least 7 different phyla that live associated with these deep sea thermal vents, we see these chemoautotrophic symbionts. And so then that builds on the community, right? So we've got the symbiosis between the eukaryote and the chemoautotrophic symbiont, bacterial symbiont, that's living right adjacent to the vent. And these crazy hard to imagine temperatures, these crazy hard to imagine concentrations of sulfur, far, far away from the reach of the sun. But because then they are there are other animals that don't particularly have symbionts, but they're allowed to live because there's an ecosystem that's created, and then they become kind of part of that. So the Riftia, the suboglanid worms that were found at these hydrothermal vents, these great big, huge gills, but they lack a gut. And that gut, the absence of the gut is made up for because they harbor these chemosynthetic bacteria. That oxidized sulfide that's prevalent and abundant in the water at that location because of the gushing out of the thermal vent to fix carbon dioxide and to make organic compounds. And so this is an example of like what we would call a holobiont. So the host, the eukaryotic host, and all of basically the symbiotic organisms that live on and within it. In this case, within it, because we have within this Riftia worm, the trophosome, which is a highly vascularized organ that houses symbiotic bacteria that provide that food, that fix the sulfur, that make the food. So in this diagram that you're looking at here, and of course, please don't spend time sketching this out. If I wanted to ask you, or when I ask you questions about this kind of relationship, I'll show you the picture. But we're looking here, basically the host in this diagram is the Riftia, is the worm, and the symbiont in yellow is the bacterial partners. And so we've got inside of mixing up ocean water and vent fluid, all sorts of pluses and minuses. So we've got sulfur oxidation and biomass production that's dependent on the relationship between these two organisms. Amazing, right? And something that you're familiar with. We've touched on this before, certainly in 2700 we talked about it. But one of the things that I want you to think about when you're thinking about symbiotic events and holobionts is we just described kind of a snapshot, a life history snapshot, the adult, say. That's the adult worm there. How does this happen in the life history cycle of the worm? How do little baby worms get their endosymbiont? And in this particular case, it seems that the trophosome is colonized by horizontally transmitted free-living bacteria that live in the surrounding environment. So we see in this life history, so we've got, I'm going to go backwards because we'll start with the adult here where my mouse is moving around. It releases its gametes into the water. The gametes meet and join and start growing. An embryo then forms a trochophore larva with its beating cilia. We saw the mustachioed Tom Selleck trochophore larva last time that are floating, free floating out in the water. And then they're infected by selected species or taxa of selected bacterial symbionts that then settles and then it grows. And the symbionts, as it grows and then the adult dies, are then released. And so we have this colonization of the trophosome from the surrounding environment. Now, that colonization from the surrounding environment isn't the only way that these patterns can happen. It's a common one. But it's not the only one, because in the last slide I mentioned that would be called or termed horizontal transmission. So the host acquiring its bacterial symbiont from the environment. Symbionts can also be vertically transmitted as a gift from your parents. So the host acquires the symbiont from the parent. And that mode of transition results in a very different co-phylogenetic or specialized to generalized continuum of relationships between the host taxon and the endosymbiont taxa or taxon. You can imagine that if you're receiving your host from your parent, that there's very likely a tighter, a closer co-phylogenetic relationship between the host taxon, in this case these these clams, these deep thermal vent clams on the left-hand side, and the taxa of bacteria that live in their gills, so tip to tip. So if we take these two phylogenies and we rotate them, so we say, okay, who's living with whom, and then we see a very tight aligning. So that's a tight co-phylogenetic relationship. And that's due to vertical transmission. If these were horizontally transmitted, just absorbed from the environment, we might expect to see some more slurring. So we would see kind of one lineage or three lineages all colonizing one, or multiple host lineages all having the same bacteria, just because it's prevalent in the water. So the mode of transmission is really tightly associated with how specialized or generalized the relationship is, the endosymbiotic relationship. And one way of describing that specialist to generalist, one measure of it, is co-phylogenetic analyses. So there's the clam and there's the endosymbiont. And we're still learning things all the time. All the time.
Yes, no, I wouldn't expect to be like so.
I love that. What? That's the WTF moment. This is Falcor, or Sebastian. The ship's on the surface. Sebastian's deep underneath. And what's happening here is that they're diving on top of a deep sea hydrothermal vent. We see the Riftia. And what they're doing is essentially what, to me, as a leaf litter, as a tropical biologist of things that live on the ground, is natural. You turn stuff over and you look underneath them. Well, what they're discovering as ROVs go down and film and send back high definition video, and the very calibrated and precise collections that can be made, is that they're discovering that these animals aren't just restricted to the surface, that they extend into cavities underneath the surface. And so the larva and potentially the endosymbionts might be connected in a way that we, before last year at this time, did not imagine at all. And so go and take a look. I love these kind of moments of discovery that are captured in these videos that are freely available. So please go and check that out. Stop what you're doing. Go and check that out. Well, maybe not. Stay with me. So the larva can either, and the endosymbiont, when the adult dies, can float out into the water column and float away passively to a new collection, or they can discharge into these cavities that are underneath the shelves. And so there are not just adults on top, but there are adults on the bottom in these hidden areas. And so there's a connectivity and potentially a reservoir of recolonization for both the endosymbiont and the larva that's happening underneath the surface, hidden from view. And one of the cool things about this, like I said, this is a study that came out like about a year ago, almost exactly. But like so many things, what's old is new again. What the authors acknowledged was the fact that there was I wonder if study that was published almost as a description, as an essay years earlier, where researchers had kind of diagrammed or sketched out this kind of a relationship and thought about it, essentially predicting what the researchers found with Sebastian and Falkor and all of those resources later on. So you can go and read that blog at Southern Fried Science there. So, symbioses on deep sea thermal vents. Amazing things that we've discovered recently, comparatively recently, and that we are still discovering more things daily. Now, other modern kind of understandings of symbioses, one that you were introduced to in 2700 last year, were these Elysia sea slugs, which live as most sea slugs or as most gastropods historically, ancestrally did, as a rasping. They have a radula that rasps, and they kind of eat the little bits of algae that they rasp up. They're a lawnmower, right? And they have these teeth on the radula that grow and grow and grow. And we'll talk about those in a second because we're going to talk about grazing. But the other way that these particular sea slugs live is via kleptoplasty, the word we mentioned earlier. As a reminder from 2700, kleptoplasty is when there are genes that are in some primary producer, like the algae, that are supporting photosynthesis, that are eating the sun, this incredible kind of miracle of life on our planet. And those genes have been acquired by the animal, by the eukaryotic animal, via horizontal gene transfer. And so there are encoded proteins for photosynthesis that occur in the genome of the slug. Now, they don't run super well on their own, and they can't provide enough nutrition on their own, but what an incredible augment. Like, as someone who has no hair on the top of their head, I would love it if I had a green sheen that I could just kind of feed off of because of my kleptoplastic algae that I keep on the top of my head. So the sequestered chloroplasts from those algae that they've grazed on are here. And the hypothesis is that the chloroplast is then partially phagocytized by that red action that I've highlighted there, by a digestive gland cell. But some of them are not, and some of them continue to live through time. Now, again, so this is a relatively contemporary relationship that we've discovered, but it is also one that we continue to discover more things about. So, for example, the Elysia snail marginata, which we thought was one and widely distributed, now it seems to be a potentially different 20 different species, maybe in 3 different genera. What other elements are there out there associated with these slugs and their kleptoplastic relationships with the chloroplasts in this case, the holobiont perspective? What does that bring to potential for drug discovery, for biological control, for all sorts of things, for my photosynthetic cap that I wish to put on my bald head? All right, that is my spiel on the symbiotic things. And so I glance down and I don't see any more questions, so we keep going. And we continue with Elysia because we're going to be talking about eventually, because we're talking about grazing and browsing. Grazing and browsing, whether it's in the ocean, as this Aristotle's lantern is here. Play. Thank you. Oh, I love that sped up video of this sea urchin kind of munching. These are, whether it's terrestrial, aquatic, or marine, these are mobile consumers of sessile prey items, often plants, that crop the tissue, typically without killing the prey. They often have specialized mouth parts that help with food consumption, like the radula in the snail, Aristotle's lantern in the sea urchin, or scleroterized jaws, as in the mandibles of your grasshoppers and your orthopterans, your insects. So, what are challenges of living like this? If you're a grazer, as this larval lepidopteran is, what are some of the challenges? Well, the food, just like with the deposit feeders the other day, similar challenges. Food is low quality and hard to digest. And so many grazers have symbiotic organisms. They are a holobiont, and they house their symbionts inside of their gut that facilitate digestion. They have long guts, increase the surface area, increase the rate of reaction. They have digestive enzymes, a suite of powerful digestive enzymes. And then from the prey's perspective, what do they have, or what do they possess that reduce the efficiency of the grazer, that potentially increase the handling time or increase the toxicity? So some are toxicity, like alkaloids, glycosides. Some, like tannins, make the proteins undigestible, and some cause altered behavior in the grazer. Now, the gastropod example that we have considered before in 2700 largely is derived from this ancestral and widely kind of adapted organ, the radula. And so we have in the mouth, we've got the odontophore that's holding the radula and all these radular teeth, this little buzzsaw that is moving out. And in addition to the digestive power provided by digestive enzymes that are produced by salivary glands that are also inside of that mouth, that's rasping its way across the surface. And you've seen videos of this if you've got an aquaria or aquaria and you've got snails within it. You've watched these radular teeth, and the specialized teeth, the number of them varies from species to species. The shape of them varies from species to species. But they're continually produced. In some taxa, you can get 1 to 5 rows of those teeth that are produced per day, that are grown and produced per day. Now, there is a small diagram in the back. You see kind of that chick, so it's like a paint scraper is what I would have said last year, but it's almost like a paired paint scraper that's scraping its way across. There's some redundancy, protection from breaking that inevitably happens. But as I mentioned, the shape of the tooth varies by the species, and it can also change depending on what species the taxon is grazing on. So what we've got here are 2 SEM, scanning electron micrographs, of 2 different teeth from Elysia viridis. So not magnifica, but one of these kind of kleptoplastic sea slugs. So for first things, take a look. The scale, although the bars sizes are the numbers are different, the scale bars are not so different. But we've got a big tooth and a little tooth. But we've got a tooth from an Elysia viridis that is collected when feeding on one species of algae, and then if it's collected on another species of algae. So there's a plasticity to the growth of radular teeth. So if we collect the sea slug, the kleptoplastic sea slug, the sea sheep oncodium, then we've got large teeth with flattened tips and prominent flanges on the outside. But if they're collected in the wild, feeding on a different species of algae, they have smaller, almost straight denticular teeth with tough, sharp cutting edges. So adaptations to the preatum that essentially reduce the handling time and increase the efficiency of feeding. Handling time and efficiency, we mentioned when we began talking about optimal foraging theory on Monday. We'll bring it up again in a few minutes. The second example, so we move from Elysia to the other kind of that gaping kind of Aristotle's lantern of the sea urchin. This fantastic organ, specialized mouth parts with teeth-like structures that can move in various directions, that eats algae primarily, but also can eat sessile animals, so it's not just a grazer. And that's kind of one of the things that we'll talk about today, is that there's fuzzy borders to these categories. Very rarely are you only one thing. I mean, we've already dealt with fuzzy borders, right? Like we've got Elysia being symbiotic and grazing. But they've got a long looping digestive system here. That has to do, as you remember from 2700, and that has to do with the fact that they're eating tough to digest things. Now, in the Aristotle's lantern, as you saw it kind of reaching out, that's these teeth here. So the long tooth that's innervated, the muscles are innervated that let it come out and consume in these incredible, I love these videos, coordinated, terrifying feeding action. Now, if you're from the West Coast, or if you spend time on the West Coast of our country, you know about sea urchins in association with kelp forests, because one of the things that we've documented, one of the changes associated with the changing conditions in the ocean, and changing conditions both abiotically, chemical temperature, but also with interruptions in food webs from the loss of predators of these urchins, historically the sea otter, that we removed because we really, really liked their fur for a long period of time. So kelp forests that ought to be filling up these oceans are often challenged by increasingly dense populations of sea urchins. And so I think you know this story. I'm going to ask you to draw out what essentially the story would look like here. So find your annotation tool and tell me what it would look like if you've been exposed to this story before. So we're going to put sea urchin abundance on the x-axis, individual sea urchins, individual kind of hungry Aristotle lanterns per meter squared, with the proportion of kelp cover. And I'll have some tea, and you tell me what kind of relationship you expect to see there. Yeah. Down. That's great. Oh, now, I think somebody just changed their guess, or they took it away. But I saw that most of you are thinking down. Some of you thought more like a negative exponential down, or you're reconsidering. This is great, because down is certainly the case. But let me overlay onto your guesses basically what we're seeing, which is that it certainly does go down, but it goes down fast. That as soon as you, so remember, and this isn't just like the number of sea urchins in an area, in a big area. This is in a meter squared. And so this is a lot of sea urchins in a meter, 10 sea urchins in a meter squared. That's a big deal. That's a big grazing pressure. And what we see is this movement from a productive kelp bed supporting dozens, hundreds, thousands of other species in these marine environments, to the sea urchin barren that is now plaguing many areas on the West Coast. Now, let me take a look at your annotation. Thank you for sharing your hypotheses with me there. I don't see any other questions yet in the chat. So we'll move from symbiosis to grazing and browsing to hunting. Tons of examples here, right? So this is from a, excuse me, apologize for that. Different kind of mouth parts associated with being a hunter in the invertebrate world. And here largely not just lots of arthropod examples. So we've got chelicerates, and we've got crustaceans, and we've got other kind of arthropods, but we've got the sand striker. So we've got annelids, ketognaths, nudibranch mollusks. But one of the common elements that would incorporate all of the adults from all of these different taxa, all of these different phyla, is that they're mobile. So they're large, and they're mobile, and that they're killing and consuming prey. They're either pursuing it, they're searching for it, they're ambushing it. And so under the heading of being a hunter, we're going to talk a little bit more about the individual strategies associated with pursuing a prey, ambushing a prey, and searching for a prey. So pursuers and ambushers. So here's kind of two different phyla examples drawn from the marine environment. We've got cephalopod mollusks that catch, chase, and subdue their prey, highly mobile prey, with organs that surround the mouth. So we've got the tentacles, and we've got all sorts of other kind of organs that we'll unpack in a second that allow them to chase, catch, and subdue. And then we've got adaptations of visual and sensory systems. So think about, for cephalopods, we've talked about other adaptations, visual and sensory, and also circulatory. Remember, we've always made the tight association between the closed circulatory system of the cephalopod with its highly mobile, predatory hunting strategy. But we've also got, we see the same similar kind of selective effects on the ketognath at a much different scale, like tiny, tiny, tiny things, but they're still responding to visual and sensory adaptations. Let's do the cephalopod first. We think about this Times article from just about 9 or 10 months ago, earlier this year. It's associated with the paper that's on the bottom right-hand side, but it just talks about and the organ, sorry, the press publication I love talks about what the crab sees before it's eaten by this cuttlefish. So it's all sorts of different, please. Adaptations of those arms, and they're chromatophores that allow them to either blend in, to be cryptic and to ambush, or to startle, or all sorts of things. I mean, some of them, I'm going to anthropomorphize for a second, but some of those look vaguely like they're going to hypnotize me into not moving away because I'm fascinated with something, and then you're consumed. But of course, an older Monterey Bay Aquarium Research Institute video of this potential like the ROV that was almost consumed by that Humboldt squid deep underwater. And then there's the ketognath at a much smaller scale. That is this little torpedo that has all sorts of sensory appendages on the areas on the front end of its body, and then this little wavy tail that allows it to move, dart, but it sits and it waits. It's essentially like, well, in a terrestrial environment, I think of a toad. A toad doesn't really chase things that don't move. It only tries to eat things that are smaller than it, that move in front of it. And that's what this ketognath is doing. So one of the voracious predators of the holoplankton, of the planktonic environment. But you've seen other ones around. If you're out in the dairy bush or in the field to the north end of the dairy bush, you might have run into mantids that are kind of relatively sedentary, somewhat concealed, that move quickly to catch and attack their prey. They often use mimicry to get close to things, and then they often have associated toxic compounds to help immobilize their prey. And that strategy has arisen multiple times independently. Some of the most mimicry examples that I think of are associated with tropical taxa that mimic ants, because mimicking an ant gets you a free pass for a lot of things, because lots of your own predators don't want to eat you, and if you can kind of hang out with a bunch of ants, you might just get to snack on ants for a while, and that's a pretty dependable food source, because one of the things that's true of our terrestrial world is that there are millions upon millions upon millions upon millions of individual ants and kilograms of ant biomass that you can eat. So this is a spider, a jumping spider, living in northwestern Costa Rica in Guanacaste at around mid-elevation rainforest, around 700 meters on a volcano from which you can see the Pacific Ocean, and it's mimicking a Pachycondylus spider there. And I mentioned these fuzzy boundaries that we expect to bump into. Remember, with filter feeding, one of the examples we talked about was the thoracopods of the krill, right? So crustaceans, filter feeders. But of course, crustaceans can do more than that. And we mentioned that a little bit when we talked about the decapod, the lobster, that the Cycliophorans were living on the mouthparts of the lobster. These are some dramatic moments that were captured in 2017 by the Econos ship that was, I think, off the coast of, like in the Pacific, kind of plus or minus around Hawaii. But it found this deep sea shrimp that impaled, and then they watched it in the bottom frame consuming this dragonfish. So fuzzy boundaries. So you can be both a filter feeder and a hunter. Fuzzy boundaries, and we've already talked about molluscan fuzzy boundaries, but even more so, you see fuzzy boundaries here where there are searchers that are actively foraging prey that are less mobile than themselves. So we see less mobile, the prey, being the bivalve mollusk here, with the more mobile searcher, the gastropod, that has ground out and has an adapted radula that is no longer just rasping and feeding upon algae, but is boring through the whole of its relative to eat the juicy contents protected within. So this is an adaptation searching being for dealing with prey that are hard to handle and hard to digest. Another fuzzy boundary would be vampire moths. This is a particular taxon that has these tiny tearing hooks on the tip of their proboscis. So this is the proboscis that kind of kept protected under here, and then usually kind of, if you think about a classic lepidopteran strategy, it's feeding on sugar water or flowers or that kind of thing. But these are feeding on blood that they're tearing and eating, tearing at a prey item's dermis and then eating the blood, lapping up the blood that emerges from it. So these are fuzzy boundaries, not just within a taxon, but across taxa in terms of how they're feeding and how they're providing themselves with nutrition. But they're all falling under essentially a mathematical, I'm going to go reductionist here, umbrella that we've been carrying around since we were talking for 2 lectures back in the first part of September in our life history strategy lectures, of the fact that there's a calculus to this, that if you are spending too much time on an individual dealing with a particularly pesky, toxic, hard to get at a mollusk shell that's just too thick and you can't rasp through it. The calculus being, if you're spending too long and too much energy, you might not acquire enough energy to replace that energy that you put into searching and dealing with and handling that organism, and you might not have enough energy left over or gained from that which you ate to reproduce. And so this overarching kind of umbrella of optimality. So there's 2 large theories that have been developed that talk about this ecologically. One of them is the marginal value theorem. So we've got the fact that animals need to maximize intake over time. And that essentially, when you're dealing with a food item that's patchy, like an individual this and an individual that, so it depends on how far apart those patches are, or those individuals are that you're preying on, and how rich it is. And so essentially this is the principle of diminishing returns, that your decision about when to leave a patch is going to be based on how far apart the next one is, and then what kind of intake you're getting, how rich the patch is. Optimal foraging theory, which is further developed by MacArthur, because MacArthur developed most of mathematical ecology in the late 60s, thinks about the fact that biologically, that ketognath, the sit and wait predator that we talked about, should continue to sit and wait until the marginal cost from moving to the next location is equal or less than the marginal cost of making that actual step. So the movement distance here, so any method for searching for that maximizes this net energy return is going to be selected for. And so this makes a prediction that if we can figure out how to measure these movement distances or body sizes or patch richnesses, we should be able to, for multiple taxa, detect where this optimality, this point is. And so, for instance, for this polychaete, this that buries itself in estuaries, researchers kind of ranked the number of different taxa that it ate. That's the kind of the rows of the table. And then the preference that it ate them is then ranked. This is based on gut content analysis, and then based on other laboratory analyses of the prey items that it had eaten. What you see is that you've got basically not a 1-to-1, 3 and 4 are reversed here, but other than that, the preference that this nondescript estuarine burying polychaete is choosing the food are essentially ranked by how much energy. So high energy prey that are easily attained, like this is scavenging a dead thing. Low handling time, low search time. It's probably being brought in by the tide and the kind of arriving and departing tide. And so there's good aligning here with our theoretical expectations and what we actually see energetically out in the field. Other examples, we see crabs that are handling different mussels of different sizes, and thinking about how profitable a larger mussel would be. Probably if it's a bigger mussel, it's to some extent it's going to be harder to get into, but it's got a lot more muscle in the mussel for you to eat once you are successfully getting that. And so they analyzed, on the graph on the left-hand side, the y-axis is the joules per second, so how much energy you're getting per unit time. And they found this exact kind of optimality predicted U-shaped curve. So a mussel that's just under 3 centimeters long seems to have the most beautiful amount of energy. And when they looked at the percent of diet that mussels of that size provided to the predator, to the crab, what they found was real corroboration that these crabs are searching optimally for the food that they're looking for. So they're maximizing energy content, minimizing handling time. Now, if you're the prey, because we're all prey of something, how do you thwart that? For a searcher, an ambusher, for a hunter, you maximize the search time. You hide. You're cryptic. You maximize the handling time. You have defenses or armor, difficult shapes to handle, and you minimize the food value. You think I'm very dilute or I'm noxious. Now, when we think about noxious materials and noxious prey, maybe as an ecologist, as a wildlife biologist, as a biodiversity scientist, as a zoologist, you start thinking about honest or not honest signals of toxicity that are common in the invertebrate world, like these coccinelle, these ladybird beetles, where we see a range of different colors, and different colors, conspicuous colors, distinctive colors that are paired with some degree of unprofitability. In this case, noxiousness or lack of palatability, warn predators against attacking them. And that strategy is not a monophyletically distributed strategy. It's like it has appeared multiple times throughout the phylogeny of animals in the world. And many of these coccinellid species possess this kind of coloration, and it's an honest signal in this case. They're releasing alkaloid-based noxious compounds, so that compounds that they have retained from the thing that they're eating, the grazing and the browsing that they're doing to prevent the hunter from eating them. And what researchers have found is that if they were to rank the toxicity here on the I'm going to say rank, but it's on the y-axis. So the highest toxic, the greatest toxicity tend to be the things that are the brightest, that are the most conspicuous, that are the honest signal to that predator. You probably want to spend your time better and more effectively by eating something that's brown or not going to be me, please. And thank you. Caramel's right. Feeding is exhausting, but there's thinking about blurred boundaries. I wanted to talk about, just as an opportunity, since a lot of this lecture seems to be shared excitement for gastropods, I hope. We'll talk about or the fact that sea slugs are incredible because they're leaves, but also they can be fish. This is a free swimming sea slug, the Phyllorelia, which looks like a fish, swims like a fish, and eats jellyfish. So it's not a grazer anymore. It's a predator. It's a mobile, searching predator. But then there are other kinds of carnivorous sea slugs, like this one that was discovered just over a year ago that has this cavernous hood. So it swims with this spatulate kind of fingered tail back here, but then the mouth, which would be up in that area, that hood that just goes, bow, 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 it's out there collecting, not filter feeding, I'm going to say because the water's not really moving through this hood. It's not a net like we saw before with the Ecoplurus, the tunicate that's nominally kind of looks somewhat similar. This is a hood that's like bringing you into a mouth. So it's taking big items and eating them. It's a hunter. And then we have other sea slugs like that are predatory as well. Fiona pinnata is one that I was taught about years ago, which is a cosmopolitan, obligate rafting nudibranch. So it lives all over the world and it's connected all over the world because if there is a bit of junk that's floating in the Pacific, it probably has this species of sea slug on it. And it's feeding on the barnacle. So it's a predator, a hunter for the filter feeding, the suspension feeding crustacean barnacle that is on. Now, what we've found out, again, like the Alicia story in the last several years, is that this is actually likely more than one species, and it is not, and there's like a, not, there is at least one of those taxa, newly thought to exist taxa that used to be kind of masquerade under this single name, that is cosmopolitan, but not all the lineages are. And so this amazing thing that, again, from the underneath, from one kept in an aquarium, kind of looks like, you can see like a shaggy, sheep-like appearance there. And then finally, thinking about all the crazy things that gastropods do, you know about cone shells, and you know about these modified radular sacs that are connected to venom glands with a bulb that kind of shoot this needle-like radula out to inject venom and paralyze the fish. And that's basically a tether and taser feeding strategy where they regurgitate indigestible material, but use their osphradium here not just to tell you kind of which species of algae you're browsing on top of, but to sense the prey because they're not doing this visually. And then the prey being fish species, right? So the adapted radula, and if you haven't seen this video about the cone snail, check it out later on. You can follow the QR code or click on the link when you're reading the PDF. And so 2 of 3 feeding strategies. We've got 3 meals, I suppose. We talked about deposit and suspension feeding on Monday. We talked about symbiosis, grazing and browsing and hunting today. And then next Monday, we're going to be talking about parasites, perhaps the most abundant, least maybe not least, but underappreciated strategy. Its abundance is underappreciated. So what to work on until then? You've got some things coming up. Your lab report is due on, the first draft is due this Friday before midnight. The case study for these 2 lectures will appear on Thursday. And if you haven't started it already, I encourage you to think about your invertebrate R assignment number 2. Please don't leave it till the end. And if you haven't already formed a group to prepare for case studies in the final, I urge you to do that. Make questions for yourselves because life is going to be easier when you do for you. And a reminder that I will be traveling again this weekend, so I'm not going to be able to respond quickly to case study or invertebrate R questions by email or to the discussion board dependably fast before Monday morning. I might be able to get some patchy signal, and I will try, but just to let you know that that is what's going to happen in my schedule. So what I'll stop recording here in a second. I'll urge you to take care of yourself, take care of those around you, and have a great rest of the week, and good luck finishing up your lab report. I will keep the chat open, and